Hi dear students, this is Dr. Mamta Jawa, your microbiology faculty. In this video module, I'm going to discuss the microbiology questions asked in the INICET May 2023 exam. So the first question goes like this. An elderly diabetic patient with immunosuppressive state presented with a skin lesion, biopsy of which showed gram-positive oval budding yeast cells with pseudohyphae. What is the most probable diagnosis? Now the buzzwords in this question are gram-positive oval budding yeast cells with pseudohyphae. So the answer is not invasive aspergillosis or mucormycosis or histoplasmosis. The answer is invasive candidiasis. Okay, this is a gram stain picture of Candida showing beautifully gram positive oval budding yeast cells. This is a schematic representation of Candida albicans. You can see these budding yeast cells with pseudo hyphae. Then chlamydospore is an example of an asexual spore which is usually formed at the ends of the hyphae. As you can see, it is thick walled, and an example of a true hypha produced by this organism is germ tube. More on this in just a bit. Now, the most common species of genus Candida infecting humans is Candida albicans. This fellow, by the way, usually is our friend. It is residing on our skin. It is in our oral cavity and the gut, and also in the genitourinary tract as a commensal. So if you examine microscopically any specimen from these sites, you will see, oval budding yeast cells and no pseudohyphae. In case you see pseudohyphae under the microscope, that should ring the bell about tissue invasion. So presence of visualization of pseudohyphae under the microscope is a very important hint of tissue invasion by the commensal. Please remember this. So because this fellow Candida albicans is a commensal, if it decides to cause disease under circumstances when the immunity is jeopardized, such infections are said to be endogenous. क्योंकि infection बाहर से तो आया नहीं, it is not acquired from outside, so it is not exogenous, it is endogenous. Now, which species of genus Candida is not yeast like? This again is an MCQ. क्या आंसर है इसका? Candida glabrata, also known as Torulopsis. Torulopsis glabrata. Please remember, this fellow does not produce pseudohyphae. It does not produce pseudohyphae unlike the other species of genus Candida. Now, Candida albicans, remember, it is said to be non-thermally dimorphic, which means at the same temperature, that is, let's say 37 degrees Celsius, that is human body temperature, it is going to be existing as the budding yeast cells, obviously, when it is just behaving like a commensal. But if it decides to cause disease and invade the tissues, then it will also produce pseudohyphae. So, because two kinds of morphologies of this fungus are existing at the same temperature, it is said to be non-thermally dimorphic. Please don't confuse this with the thermally dimorphic fungi like histoplasma, blastomyces, वगैरह वगैरह. ठीक है. Now, Candida albicans क्या infections cause करता है? Remember oral thrush, then esophageal candidiasis, which by the way is an age-defining illness. So number 1, 2. Then thirdly, remember vaginal candidiasis, which is seen in usually diabetic females. It is characterized initially by a watery vaginal discharge, which later on becomes thick and curd-like. The patient has got intense pruritus, and remember, vaginal pH is not altered in vaginal candidiasis. Vaginal pH is altered where? In trichomoniasis, okay, and bacterial vaginosis. Then further infections you can remember as I2CU. Now I2 में पहला I क्या है? Infective endocarditis. Candida albicans can really be a cause of it. And then intertrigo, which is nothing but infection of that area or those areas of the skin where skin forms folds like some mammary region and axillary region, वगैरह वगैरह. Then C stands for what? Candidemia, a deadly infection characterized by bloodstream invasion by this fungus. Then U stands for UTI. Now, for diagnosis, sample collection has to be done depending on the type of presentation. So you can go ahead with urine collection, blood collection, वगैरह वगैरह. Then microscopic examination में आती हैं दो चीजें, wet mount and gram stain, which will show gram, which will show the budding yeast cells like this. wet mount पे ऐसा दिखेगा. gram stain पे बस एक additional चीज आपको दिखेगी कि you will see the color gram positive ऐसे दिखेंगे. and in case अगर आपको pseudo hyphae भी दिखते हैं, चाहे wet mount पे दिखे या gram stain पे दिखे, remember it is a very important hint for what tissue invasion happening. Then culture on STA, Sabrot, Dextrozagar, which by the way is the most commonly used fungal culture medium. इस पे भी question क्या आता है इसका pH के बारे में. pH average is 5.6 and the range of pH is 4 to 6. Now on this medium SDA, the colonies of Canada albicans look like this. क्रीमी पेस्टी कॉलोनीज, टूथपेस्ट जैसी दिखने वाली सफेद कॉलोनीज. एंड लेट्स से अगर आपने ये कॉलोनी आपको मिली, यू कैन प्रोसीड विद द जर्म ट्यूब टेस्ट, वेयर बाय यू टेक द कॉलोनीज, मिक्स इट विद ह्यूमन और शीप सीरम एंड इंक्यूबेट फॉर 2 hours. ठीक है. एंड देन व्हेन यू एग्जामिन अंडर द माइक्रोस्कोप, you see this tubular projection or emanation arising from this parent cell or the yeast cell. This is nothing but germ tube. Germ tube formation is also known as Raynaud's Brody phenomenon. Remember this. Then growth on cornmeal agar can also be done to demonstrate the formation of chlamydospores, which I talked about before. And then growth on CHROMagar. अब CHROMagar के बारे में क्वेश्चन INICET में ही आ चुका है. They gave a clinical vignette and then they just ask कि which of the following media will you use to arrive at the name of the species. तो आंसर इस क्वेश्चन का CHROMagar था. This question was asked in another INICET exam. ठीक है, I think it was November 23, and the same question with options and all, I have an explanation I have posted on my Insta handle, and the that question का link also I am going to post in the description of this video, तो उसको भी please question को देख लीजिएगा. Now differentiation of Canada species by isolation on CHROMagar Canada medium. So remember guys, आपको बस 3 इसमें चीजें याद रखनी है, 3 स्पीशीज के नाम याद रखने हैं. कैनेडा एल्बिकन्स, जिसकी कॉलोनीज कौन सी होती हैं, जिसकी कॉलोनीज होती हैं ग्रीन कलर की. ओके, सो ग्रीन कॉलोनीज. So guys, green colonies are of Canada albicans. ठीक है, green colonies are of albicans. The blue-gray colonies are of tropicalis, and the large rough pale pink colony. इतना लंबा description, large rough pale pink, ये वाली colony ये किसकी है? इतना खड़ूस सा सूखी सूखी सी colony दिख रही है. Remember खड़ूस and cruci, they are somewhat similar sounding words. ठीक है, this is how I used to remember. ठीक है, तो ये 3 स्पीशीज के नाम और इनकी कलर ऑफ कॉलोनीज आप याद रखो. दीज आर द 3 स्पीशीज व्हिच कैन बी रिलायबली recognized on this medium. This is very important about CHROMagar. Now I have a hunch कि अगर उन्होंने CHROMagar पूछा है to कि भाई आप स्पीशीज पता करने के लिए कौन सा मीडियम यूज करोगे, then in a future question which I can speculate is what else you can do to speciate Canada. उसका आंसर है carbohydrate fermentation and assimilation test. So these are the tests for what? To do speciation, to arrive at the specific species of genus Canada. Then now finally guys, now if we talk about the treatment aspects, remember for cutaneous candidiasis the drug of choice is topical azoles. For esophageal and vulvovaginal candidiasis, remember it is oral fluconazole or caspofungins can be given. And for disseminated candidiasis, remember amphotericin B, preferably liposomal amphotericin B and caspofungins. And another MCQ क्या पूछा जाता है इस टॉपिक पर? Species refractory to azoles. Remember Candida glabrata, which is not going to produce pseudohyphae as I mentioned before, usko yaad rakho. And Candida cruci, the khadoos one, both of these are refractory to azoles. So this finishes this question. Now let us proceed with the next question. Identify the infection as per the morphology of the organism in this question. In this image, look at this. तो क्या दिख रहा है? You can see filamentous structures, septae दिख रहे हैं, और acute angled branching है, for example यहाँ पे है. ठीक है. Now it is definitely obviously not mucormycosis or histoplasmosis or blastomycosis. Straightforward answer क्या है? Aspergillosis. Now Aspergillosis, remember guys, Aspergillosis, if I write the name Aspergillus, remember it is a hyaline mold, a hyaline fungus. And if I highlight A and S of its name, A से याद रखो acute angled or dichotomous branching, acute angled or dichotomous branching. Now dichotomy का literal meaning क्या होता है? Meaning twofold, classification into two opposed parts. तो dichotomous branching का यही मतलब है, this is hypha which is septate and it branches at it acute angle, so this is known as dichotomous branching. ठीक है, then S stands for what? Septate present. So septate hyphae present होते हैं इस फंगस में, एस्परगिलस आप ऐसे याद रख लो. Then what are the risk factors? Now the most important risk factor is the use of steroids. ये सबसे important है, most important. Then the second risk factor is pertaining to those cells which are going to play the most important part in immunity against disease by this fungus, that is the neutrophils. तो neutrophils का या तो decreased number हो या फिर इनका dysfunction. Now this is a very important reminder for the fact that Aspergillosis, unlike candidiasis and cryptococcosis, is not a common fungal infection in HIV infected individuals, क्योंकि HIV infected individuals में neutrophil count जो है वो end तक end में खराब होता है. So उनमें neutropenia beginning में नहीं होता. HIV में problem कहाँ पे हो रही है? CD4 cells, that is the T helper cells का count decrease हो रहा है. ठीक है, so that is why Aspergillus जिसके लिए if we talk about a proper immune response, neutrophils are such an important immune cells. तो वो तो नहीं हो रहा HIV वालों में. So please remember this aspergillosis, this fungal infection is not at all a common fungal infection in HIV infected individuals until the very late stages of HIV infection. Please remember this. ठीक है, then third important risk factor is and some लंग कंडीशन जो लॉन्ग टाइम से चले जा रही है. लेट्स से अंडरलाइन कुछ निमोनिया है जो ठीक नहीं हो रहा है, COPD, sarcoidosis, all these conditions if they are there in some patient, then anti-tumor necrosis factor therapy, this also is a risk factor. Now types of aspergillosis quickly हम देख लेते हैं. Firstly ABPA, allergic bronchopulmonary aspergillosis. अब भाई इतना लंबा नाम, तो स्पीशीज का नाम भी थोड़ा लंबा ही है, एस्परगिलस फ्यूमिगेटस. फ्यूमिगेटस से फ्यूमी से फ्यूरियस याद करो, तो फ्यूरियस गुस्सा आना, तो लंबा वाला जो नाम है ना इस बीमारी का उसको ऐसे ही आप याद रख लो. 3 इंपॉर्टेंट स्पीशीज जनरली आपको एस्परगिलस के पता होने चाहिए, फ्यूमिगेटस, फ्लेवस एंड नाइगर. सबसे लंबा नाम फ्यूमिगेटस है. एबीपीए में भी मोस्ट कॉमनली implicated species is this one only, and this condition, allergic bronchopulmonary aspergillosis, is usually seen in patients with cystic fibrosis and those with asthma. और इसमें क्या होगा, the patient is going to have elevated IgE antibodies against the species which is incriminated in this in condition ABPA. Also in this condition the patient is going to have wheezing and central bronchiectasis. These are also MCQs. ठीक है, then aspergilloma, also known as क्या? Also known as fungal ball. Now what does this mean? This means, let's say there is a cavity in the lung. Let's say this circle is a cavity. Now the two important causes of cavity in lung formation are TB and cancer. तो क्या होगा, in this pre-existing cavity aspergillus will just go and colonize this cavity. So that is nothing but fungal ball and it is not going to invade the cavity. So remember this entity is non-invasive. ठीक है, non-invasive. Then we have got invasive aspergillosis whereby angio invasion occurs, that is invasion of the blood vessels. And angio invasion कब होगा? When there is neutropenia, profound neutropenia. So angio invasion की वजह से क्या होगा, the blood supply will be cut off to that very tissue, resulting in infarction and necrosis. Please remember. Then keratomycosis can be seen in farmers during the harvesting season because of injury to the eye using the splinter. This fungus, by the way, aspergillus, it is just like God, it is omnipresent everywhere. ठीक है, so by trauma it can enter the eye and the farmer can land up with keratomycosis. Then otomycosis is important also क्योंकि this is the overall most common infection. Overall this is the most common infection by this fungus, that is aspergillus. और इसका most common species causative कौन सा है? Aspergillus niger. Now look at this guys, this is how the colonies of Aspergillus niger look like. Niger का मतलब ही क्या होता है? Black. Remember this. ठीक है, then aflatoxicosis क्या है? This is not an infection, it is intoxication. Afla कहाँ से आया? It comes from what? Aspergillus flavus, which produce what? aflatoxins, which are hepatotoxic, So let's say there are some grains which get contaminated with the fungus, which obviously we can't see. Water sets in. So what will happen? There will be, a germination of the spores, and consumption of such contaminated grains is going to lead to what? Consumption of these aflatoxins. In case, obviously, the infecting fungus is Aspergillus flavus. Okay. Now, lab diagnosis, the mainstay of diagnosis is not culture, it is biopsy. Because remember, this fungus is a saprophyte also. So just because it grows in culture is not enough for us to conclude that it is the causative agent of infection also. So biopsy is very important, and demonstration of the fungus by, for example, tissue stains, fungal tissue stains like HNE. Culture on STA can be done, but as I said, it is not the mainstay of diagnosis. Okay, it is not the mainstay. Please remember. Okay. Now this is a very beautiful picture of Aspergillus fumigators showing the septate hyphae. So I would want all of you to please note these two black colored arrows. The bigger black colored arrow is pointing towards the septa, and the smaller black colored arrow is showing you that typical, acute angled or dichotomous branching. Now, antigen detection can be done. For example, galactomannan. Antigen detection can be done. Then antibody detection against the antigens of this fungus can also be done by ELISA. Then beta 1,3-D glucan assay can be done. Now, it is also known as what? It is also known as 1,3 beta D glucan. 1,3 beta D glucan. But on purpose, I have preferred the name beta 1,3-D glucan. You know why? Because B for beta, B for broad. So remember, beta 1,3-D glucan hai kya? It is nothing but a cell wall polysaccharide present in a broad range of fungi. So a broad range of fungal infections can be diagnosed by detecting this very polysaccharide. So this beta 1,3-D glucan, which is present in a broad range of broad range of fungi, it is not seen in the following. Remember, the exceptions ke naam also are beginning with a B. So what are the exceptions? B, C, Z. So B stands for blastomyces, dermatitis. C stands for cryptococcus neoformans, and Z stands for zygomycetes. ठीक है? So please remember these exceptions. Then treatment के लिए, now invasive aspergillosis के लिए what we can go for? Invasive aspergillosis के लिए the voracious drug to be used, that is voriconazole. So invasive disease is something voracious, खतरनाक, तो voriconazole. ABPA के लिए क्या कर सकते हैं? itraconazole. ठीक है? तो ABPA एलर्जिक ब्रोंको पल्मोनरी एस्परगिलोसिस, लंग कंडीशन. ठीक है? तो इत्र मींस सेंट. तो इस तरह से याद कर लो, ABPA, इत्राकोनाजोल, एंड क्रॉनिक पल्मोनरी एस्परगिलोसिस में दोनों को ऐड कर दो, वोरिकोनाजोल प्लस इत्राकोनाजोल. तो ऊपर वाले दो ड्रग्स टुगेदर. देन सिंगल एस्परगिलोमा के लिए एक्चुअली सर्जरी इज इंडिकेटेड. एंड टॉकिंग अबाउट प्रोफाइलैक्सिस, पोसाकोनाजोल. Now, next question. What is the diagnosis based on the given image? Now, let me just zoom the image for all of you. So you can see, what is this arrow pointing towards? You can see hyphae. There are no septae, and they don't have a very uniform diameter. They are looking, they're broad, and they're ribbon-like. They're broader than the Aspergillus hyphae, which we saw before. Okay? So what is this? This is definitely not Aspergillosis. This is not Nocardiosis or Fusariosis, it is Mucormycosis. Okay? Now, this is a picture of Mucormycosis caused by mucor species. You can note the necrotic area involving the nose and the face. So remember, Mucormycosis, which created a havoc during the COVID era also. Now, it belongs to the mucormycosis is an infection caused by the fungi belonging to the class Zygomycetes, also known as kya? Phycomycetes. Now, A and Z are the first and last alphabets in English. So I would want all of you to remember that A stands for aseptate and Z stands for zygomycetes. So remember zygomycetes के पास aseptate hyphae होते हैं, and these hyphae are like this. They are branching at a right angle or obtuse angle, and they are broad ribble-like hyphae. Please remember this. And the fungi belonging to this class also, just like Aspergillus, are angio-invasive fungi. Angio-invasive fungi. And the two important risk factors for mucormycosis are, remember, DD or demand draft, that is diabetic ketoacidosis and desferrioxamine therapy. Desferrioxamine therapy. Now, what is the mainstay of treatment of mucormycosis? By the way, the most deadly disease is rhinocerebral mucormycosis. The mainstay of treatment is not amphotericin B, it is surgical debridement. Obviously you will start amphotericin B, but that is not the mainstay of treatment. Mainstay of treatment is a surgical debridement, plus additionally you will start amphotericin B. Next question. A cattle breeder in South India presented with the following lesion. HP is also given. What is the likely pathogen leading to this? So you can see this a kind of lesion in the nose, and the patient hails from South India, and uh, the microscopy is also given to you. So this is a clear-cut case of what? Infection with Rhinosporidium seaberry, because it looks like nasal rhinosporidiosis. The most common clinical presentation of infection with Rhinosporidium seaberry. It is not Klebsiella rhinoceromatis infection or Staph aureus infection. So rhinosporidiosis, previously it was fungus. Okay. So rhinosporidiosis, by the way, it's caused by Rhinosporidium seaberry. Rhinosporidium seaberry. Previously, it was a fungus. Now it is not. Then what it is? Now it is regarded as what? It is an aquatic protestin parasite. Aquatic protestin parasite or a mesomycetazone. Mesomycetazone. This MCQ also used to be asked a few years back. Now, what is the peculiar feature of this organism? It is non-cultivable. An old question on this organism is that which of the following fungi are non-cultivable? It was asked in the era when it was deemed as a fungus. So this organism, let's not call it a fungus anymore. This organism is non-cultivable. Now, maximum cases of rhinosporidiosis are from what? Sri Lanka. And remember, the most common presentation of infection with this fungus is nasal polyp, जिसकी photo हमने question में देख ली है. Then what about India? India may please remember the areas from where the cases are reported. Remember, Kota, K-O-T-A. K for what? K for Kerala, O for Odisha, T for Tamil Nadu, and A for Andhra Pradesh. So remember Kota. Now, the most common clinical form, as I said, is nasal rhinosporidiosis. Other sides of the body can also be infected, which can be remembered by remembering big leg, where B stands for kya? Bronchus. L stands for larynx. E stands for ears. G stands for genitalia. And the source of infection is stagnant water, and the mode of infection is inhalation of the spores present in the stagnant dirty water of ponds, lakes, etc., etc. And as we saw in the question also, you can see this microscopic picture. So what are these? These are nothing but spherules containing the endospores. So this is a schematic representation where you can see what is this. This bigger circle is nothing but a spherule, and this spherule is lodging inside what? The endospores. So what this spherule is known as endosporulating. Endosporulating spherule it is. And the endospores inside are 6 to 9 microns in size, and the endosporulating spherule can be in size up to 350 microns. And this again is an MCQ. Best stain for this spherule is Mayer's Muci carmine stain. Okay, Mayer's Muci carmine stain. Now, treatment, the mainstay of treatment is surgical excision. As far as drugs are concerned, dapsone can be given, but recurrences are common. Then next question. A child presented with a facial rash with a slap cheek appearance, which other condition is caused by the probable causative agent of this rash? So slap cheek appearance, they already have mentioned. So what you should think of? You should think of, this is how, by the way, the slap cheek appearance looks like. So what you should think of? You should think of infection with what? Parvovirus. Parvovirus B19. Parvo itself means small, so remember it's the smallest medically significant virus infecting humans, and it is an example of a DNA virus. If you find a hard time remembering the names of the DNA and the RNA viruses and you don't understand what is meant by single-stranded, double-stranded virus, I did a video on the same. It's not a lengthy video. The link of the video I'll post in the description. Please have a look. So guys, parvovirus B19, it's a DNA virus. Remember, it's a DNA virus. ये बेचारा इतना छोटा है कि भगवान ने इसको 2 strands भी नहीं दिए DNA के. Remember, it has got single stranded DNA as its genome. Otherwise, the remaining DNA viruses have got double stranded DNA as the genome. Now, what is the receptor? The receptor is the P blood group antigen present on the developing RBCs, that is the erythroblast. That is the erythroblast. Now, the peculiar feature about this about the infection by this virus is that the infection is usually self-limiting, and if in case immunity is jeopardized because of some reason, then it becomes a problem. Now, as far as the diseases are concerned, in kids, it causes what? It causes erythema infectiosum, which is characterized by this rash more prominent on the cheeks, and this is followed by a less prominent lacy reticular rash on the body of the kid. So remember, erythema infectiosum, also known as kya? Fifth disease. A for, then adults mein kya hoga? A for adults, A for what? Arthritis. This arthritis, by the way, is more commonly seen in females than males, and it usually affects the small joints, mimicking what? Rheumatoid arthritis. Okay. So A for adults, A for arthritis. People with hemolytic anemias like thalassemia, sickle cell anemia, they already are, they are endowed with the loss of RBCs. And as I said, this infection, although is self-limiting, but if imagine these people get infected with this virus whose target is the erythroblast, these people will develop an aplastic crisis. But because the viral infection itself is self-limiting, it is going to be transient. So in people with hemolytic anemias, it causes transient aplastic crisis. ठीक है, transient aplastic crisis. And immunocompromised people like HIV, it will cause what? Pure red cell aplasia. Pure red cell aplasia, because the infection is not going to be cleared because of the jeopardized immunity of these individuals. So please remember this. Then next question. An adult female presented with foul smelling discharge per vagina, pap smear images given. What is the correct description shown on the pap? So epithelial cells with cocobacilli stuck on the surface, macrophage with engulfed bacteria, squamocolumnar metaplasia or trichomonas. Now let me zoom the picture. Now this is a clear picture of what? This is nothing but a clue cell, that is the epithelial cells with cocobacilli stuck on the surface, and that is the answer to this particular question. Now look at this, guys. So generally in vagina, the lactobacilli are also known as the doderline bacilli. They are present in abundance, and they maintain the acidic pH of a vagina. Now, there's a condition called as bacterial vaginosis, which is not an infection but an alteration in the vaginal flora. Now, this condition is characterized by what? This condition is characterized by an increase in number of organisms like Gardnerella vaginalis. There's decreased number of lactobacilli in this condition. Okay? So lactobacilli become less. Now, there's an increase in number of most commonly Gardnerella vaginalis. Remember this. And anaerobes like Peptostreptococcus, Mobile uncus, okay? Mobile uncus and Mycoplasma hominis. So all these organisms, these 4 organisms are going to increase in number, and there's going to be a diminution in the number of lactobacilli, which look like this. These are gram-positive bacilli. Okay? Gram-positive bacilli. Now, lactobacilli have been named after Sir Albert Doderlein, and that is why they're also known as Doderlein bacilli. Now, what are the risk factors for bacterial vaginosis? Why this alteration in vaginal flora occurs? Number one, if there is coexisting other infections, like for example, HIV infection, chlamydia, trachomatis infection, or gonococcus infection. So that is the first risk factor. Then the second risk factor is recent unprotected vaginal intercourse. Then the third risk factor is vaginal douching. And the fourth risk factor is premature rupture of membranes and preterm labor. Now, diagnosis में भी इसके question आता है. There is something called as Amsel's criteria, whereby we have got four features. If 3 out of 4 criteria are present, the patient is labeled to be having bacterial vaginosis. Now, the very first feature is about the discharge which the patients of bacterial vaginosis present with. So, how is the discharge? It is profuse, thin, that is watery, meaning to say it has got low viscosity, profuse, thin, white colored, homogeneous vaginal discharge uniformly coated on the vaginal wall. The second point is about the pH of this discharge. The pH of the discharge is more than 4.5. So remember, the pH is altered. The normal vaginal pH is acidic, that is less than 4.5. In this condition, it is altered. In which other condition it is also altered, as I said before, in trichomoniasis also. But in vaginal candidiasis, it is not altered. This again is an MCQ. Okay? Then thirdly, clue cells, that is the vaginal epithelial cells coated with the cocobacilli, and these cells have a granular appearance. Let us have a look at the question wali picture once again. So this vaginal epithelial cell is having this granular appearance because of the increase in number of the all the organisms which are listed. And even though this picture is showing just one vaginal epithelial cell, when multiple cells are seen under the microscope, the borders also seem to be indistinct. Okay. So the third criteria under Amsel's criteria is clue cells. Then the fourth criteria is whiff test, positive whiff amine test. Whiff amine test is positive. Now, whiff amine test is what? You take the vaginal discharge and mix with 10% KOH. If you are able to appreciate a fishy odor, that is because of the amines. So if this odor is appreciated, we label this test as a positive whiff amine test. So it is because of the volatile amines like trimethylamine. If 3 out of 4 criteria are present, yes, we can label that patient as a patient of bacterial vaginosis. Then there is something called as Newen score. इस पे भी अभी recent NEET PG में question आया है. So basically this is a scoring system for the diagnosis of bacterial vaginosis. और इसमें होता क्या है? इसमें the counting is done of certain bacteria. Counting of the following bacteria: Gardnerella vaginalis, Mobiluncus, and Lactobacilli. All these bacteria, their numbers are counted in a gram stain smear of the vaginal discharge, and a score of 7 or more is said to be diagnostic. A score of 7 or more is said to be diagnostic. This was asked in NEET PG 2024, which was held on 11th August. ठीक है. Now इस पे भी क्वेश्चन आया हुआ है ड्रग ऑफ चॉइस पे. द ड्रग ऑफ चॉइस गाइस ओवर हियर इज ओरल मेट्रोनिडाजोल. एंड इस पे एक और क्वेश्चन नीट पीजी का ये है. They had asked a question and of a number of genital infections and asked the treatment of partner is not done in which of these cases. उसका आंसर था bacterial vaginosis. So इसकी डोज क्या है? 500 mg, 500 mg BD given for 7 days. Please remember this also MCQ है ये. Next question, what is the most likely organism in accordance with the image shown? तो पहले image देख लेते हैं. Now look at this. So you can see, okay, cytoplasmic membrane के ये proteins हैं. This is the membrane. This is cytoplasmic membrane. Periplasmic space, outer membrane. They have mentioned periplasmic space, outer membrane. What more do we want? It's a gram-negative organism. They have also mentioned lipo-oligosaccharide. There's a difference between lipopolysaccharide and lipo-oligosaccharide. Tiny difference. I'll tell you in just a bit. Then pilus. Pilus again is something which is there in gram-negative bacteria for attaching to the host cell receptors. And capsule is an optional structure, can be seen in both gram-positive and gram-negative. Then outer membrane proteins, and these are the membrane phospholipids. ठीक है। नाउ स्टेफाइलोकोकस ग्राम पॉजिटिव है, रूल आउट हो गया। ठीक है, बचे कौन से, बीसीडी, तीनों के तीनों नेसीरिया, मेनिंजाइटिस, इकोलाई, सीडोमोनास सब ग्राम नेगेटिव तो हैं, लेकिन क्वेश्चन में फिर हिंट क्या है हमारे लिए, लाइपो ऑलिगोसैकेराइड ना, लाइपो ऑलिगोसैकेराइड जो है दैट इज प्रेजेंट इन नेसीरिया मेनिंजाइटिस। Now, lipooligosaccharide is nothing but endotoxin. Endotoxin का यूजुअल और पर्यायवाची क्या होता है, लाइपोपॉलीसैकेराइड। ठीक है, तो इकोलाई एंड स्यूडोमोनास में लाइपोपॉलीसैकेराइड होता है, निसीरिया में क्या होता है, द एंडोटॉक्सिन का जो आउटर पॉलीसैकेराइड वाला पार्ट होता है ना, दैट इज स्मॉलर, दैट इज व्हाई इट इज रेफर्ड टू एज ऑलिगोसैकेराइड। तो यहाँ पे आंसर क्या है, निसीरिया मेनिंजाइटिस, नॉट जस्ट इन निसीरिया मेनिंजाइटिस, इन निसीरिया गोनोरिया दैट इज गोनोकोकस आल्सो लाइपो ऑलिगोसैकेराइड इज प्रेजेंट। सो दिस इज द बिगेस्ट क्लिंचर फॉर आवर आंसर, लाइपो ऑलिगोसैकेराइड मेंशन्ड इन दिस क्वेश्चन। ठीक है, तो ये दिस इज द बिगर व्यू of the picture. Now, this is a schematic representation of endotoxin or lipopolysaccharide, as I said. ठीक है, so inside to outside अगर जाएं तो toxic moiety is lipid A. So this endotoxin is present where it is present in the outer membrane of gram negative bacteria, GNBs, gram negative bacteria. ठीक है, सो अंदर से बाहर अगर हम जाएंगे, सबसे अंदर क्या कौन होता है, लिपिड है, दिस इज द टॉक्सिक पार्ट, दिस इज एन एमसीक्यू इन इटसेल्फ। द एंडोटॉक्सिक शॉक इज बिकॉज ऑफ एंडोटॉक्सिन और एलपीएस, एंड हु इज द विलन और द culprit or the toxic moiety, it is the lipid A. So you can see all these fatty acids dangling from lipid A. Lipid A itself is a disaccharide diphosphate. इसके बाहर क्या होगा, कोर पॉलीसैकेराइड दिस, और इसके भी बाहर होगा एक और प्रकार का पॉलीसैकेराइड, ठीक है, which is lengthier because, and that is why it is not known as lipopolysaccharide. Now this outer polysaccharide, this is the outer polysaccharide. So this is the first part is lipidase, second one is core polysaccharide, let me write it core PS, third one is outer polysaccharide. Outer polysaccharide is also known as somatic antigen or O antigen, and this is the basis of what, this is the basis of serotyping. Now this outer polysaccharide which we are labeling as O antigen is shorter in size in Neisseria meningitis and Neisseria gonorrhoeae. That is why it is known as lipo-oligosaccharide. Please remember this. And do you know any gram-positive organism, gram-positive bacterium which has what endotoxin in its structure? It is one of my most favorite bacterium because it is gram-positive. Let me write the name in blue. It is Listeria monocytogenes. This itself is an MCQ, guys. Please remember this. Now the action of endotoxin, quickly इस पे भी questions आते हैं. So as I said, endotoxin, especially the lipid A, which is the culprit, it is going to activate, remember, MCT. M से macrophages, C से complement, T से tissue factor. When macrophages are going to be activated after endotoxin binds to them, there is going to be production of interleukin 10, TNF and नाइट्रिक ऑक्साइड. रिमेंबर इट एज I10 वाली गाड़ी, I10. I से क्या हो गया, इंटरल्यूकिन 1, ठीक है, एंड अगर मैं इसको 10 को ऐसे लिख दूं, TEN, T से ट्यूमर नेक्रोसिस फैक्टर और N से नाइट्रिक ऑक्साइड. इंटरल्यूकिन 1 यू ऑल नो इट्स एन एंडोजीनस पायरोजन, ठीक है. देन नाइट्रिक ऑक्साइड कॉजेज हाइपोटेंशन. TNF बीच में लिखा हुआ है, तो रिमेंबर TNF बीच में प्रेजेंट है, वो इधर का और इधर का फीवर हाइपरटेंशन दोनों फीचर्स करा रहा है. बाय द वे, साइटोटॉक्सिन्स पे भी अभी रिसेंट नीट पीजी में ही क्वेश्चन आया. मैंने साइटोकाइन्स का अलग से वीडियो पहले किया था, उसका डिस्क्रिप्शन, उसका लिंक भी मैं डिस्क्रिप्शन ऑफ दिस वीडियो डाल दूंगी, प्लीज हैव अ लुक. नाउ कॉम्प्लीमेंट जब एक्टिवेट होगा तो C3A C5A प्रोड्यूस होंगे, विच यू ऑल नो आर एनाफाइलाटॉक्सिन्स. दे विल कॉज हाइपोटेंशन एडिमा. Hypertension edema not only will be caused by C3A but C5A also, and C5A, this again is an MCQ, it's a chemotactic agent for neutrophils. Then tissue factor भी activate होगा तो end result क्या होगा, activation of coagulation cascade, that is DIC. Finally, now next question guys. अब इसमें क्या है ना, there were two set of students जिन्होंने अलग अलग बातें बोली. तो क्वेश्चन की स्टेम तो यही है, ऑप्शंस भी यही हैं, बट पिक्चर ये आई थी या दूसरी आई थी, आई विल डिस्कस बोथ. सो द फर्स्ट सेट ऑफ स्टूडेंट्स टॉक्ड अबाउट दिस वेरी पिक्चर. ठीक है, तो क्वेश्चन क्या है, an elderly presented with the features of meningitis. Caesar sample was collected and subjected to gram stain shown in the image, which of the following is true. So let me just zoom this image. So this gram stain shows you what, it shows you gram positive diplococci with a halo around them, which is going in favor of what, pneumococcus. ठीक है, pneumococcus. Now another set of students told me the same question, but उन्होंने क्या बोला, क्या पिक्चर आई थी, इस प्रकार की पिक्चर आई थी. What is this? These are gram positive, rods, and remember it's an elderly patient. तो हम इसमें क्या सस्पेक्ट करेंगे? नाउ पहले वाले सिनारियो में अगर जाएं तो वो तो स्ट्रेट न्यूमोकोकस ही है. सो व्हाट दे आर आस्किंग about the motility aspects of the organism. Motile at 37 and motile at 37 and 22 both. 37 is human body temperature, 22 is the environmental temperature. Non-motile at both, motile at 37, non-motile at 22, and non-motile at 37 and motile at 22. Now pneumococcus, remember as a general rule, all the cocci are non-motile. तो अगर pneumococcus की फोटो आई थी तो फिर इसका आंसर क्या बनेगा? non-motile at 37 and also at 22. लेकिन मान लो अगर पिक्चर ये वाली थी, ग्राम पॉजिटिव रॉड्स शो कर रहे थे, एंड it was an elderly patient. So what which organism should it be, which is probably the most common agent in elderly? Of course elderly में देखो, most common agents of meningitis in elderly people are enteric gram negative organisms. but यहाँ जो फोटो दे रखी है वो ग्राम पॉजिटिव organisms की है, एंड ग्राम पॉजिटिव रॉड्स में एक लिस्टीरिया मोनोसाइटोजीन्स ऐसा ऑर्गेनिज्म है which can cause meningitis in extremes of ages. ठीक है, तो अगर अगर ये वाली पिक्चर आई थी तो हमारा आंसर क्या बनेगा? लिस्टीरिया मोनोसाइटोजीन्स, रिमेंबर इट्स अ फूड बॉर्न पैथोजन, इट्स अ सैप्रोफाइट. एंड मैं कैसे याद कराती हूँ, कैसे पढ़ोगे इसको? जब ये सैप्रोफाइट है, एनवायरमेंट में प्रेजेंट है, तो ये इधर से उधर घूम रहा है, भ्रमण कर रहा है. बट द मोमेंट इट एंटर्स द ह्यूमन बॉडी, एंड बिकॉज दिस ऑर्गेनिज्म वांट्स टू डिस्टर्ब सम ह्यूमन बीइंग, इट वुड लाइक टू स्टे इन द बॉडी ऑफ ह्यूमन बीइंग फॉर सम टाइम एंड नॉट मूव हियर एंड देयर. तो जब ये एनवायरमेंट में घूम रहा है तो एनवायरमेंटल टेम्परेचर पे इसको मोटाइल बना दो और 37 पे नॉन मोटाइल. सो द आंसर इज मोटाइल इज नॉन मोटाइल एट 37 एंड मोटाइल एट 22 डिग्री सेल्सियस. अब ये तो याद करने का तरीका हो गया. अब रीजन क्या होता है? एक्चुअली रीजन ये होता है कि जो इसका इसका जो फ्लैजुलर जीन होती है ना, द फ्लैजुलर प्रोटीन, दैट द एक्सप्रेशन ऑफ दैट जीन इज टेम्परेचर डिपेंडेंट. द जीन फॉर फ्लैजुलर प्रोटीन इज एक्सप्रेस्ड एट environmental temperature, it is not expressed at 37. That is the actual reason. Please remember this. Next question. Critical care specialists have grown organisms in different cultures and observed resistance to carbapenems, aminoglycosides, polymyxins, and susceptible to cotrimoxazole, tecarcillin, and clavulanic acid. What is the most likely organism? तो ये थोड़ा सा अजीब सा क्वेश्चन एक आया था. Now the answer of this is Stenotrophomonas maltophilia. ठीक है, अब I had posted on Instagram a post regarding the intrinsic resistance of certain important organisms. उनमें से एक मैंने ये भी पिक कर लिया था because इस पे क्वेश्चन आया था. So let me just show you those slides. वो Instagram post भी I will the link of that post I will share in the description of this video. I'm going to post a lot of links, quite a few links actually in the description. Please have a look. वो सारी मेहनत आप लोगों के लिए है, ठीक है, उसको प्लीज देख लेना. नाउ एग्जाम्पल्स ऑफ इंट्रिंसिक एंटीमाइक्रोबियल रेजिस्टेंस. स्टेनोट्रोफोमोनास माल्टोफिलिया. देखो, स्टेनो मतलब नैरो, ट्रोफो मतलब वन हु फीड्स, ठीक है, मोनास मतलब यूनिट. माल्टोफिलिया, माल्टो से माल्ट सोच लो, फिलिया से मतलब फिलिया मतलब टू हैव एफिनिटी और अट्रैक्शन और बिफ्रेंडिंग समवन. ठीक है, सो ऐसे याद करो. दीज आर नैरो बैक्टीरिया दैट नीड फ्यू सबस्ट्रेट्स फॉर ग्रोथ. सो जीनस स्टेनोट्रोफोमोनास के बारे में हम ये बात बोल रहे हैं. देन द स्पीशीज माल्टोफिलिया इज अ फ्रेंड ऑफ माल्ट. तो मतलब नाम से तुम्हें क्या पता चल रहा है? द नेम सेज माल्ट मतलब कार्बोहाइड्रेट. इसका मतलब इसको और ज्यादा कार्बोहाइड्रेट्स नहीं चाहिए, नो मोर कार्ब्स. सो इट इज रेजिस्टेंट टू कार्बापेनम्स. इट विल नॉट एक्सेप्ट कार्बापेनम्स. दिस इज द ट्रिक टू रिमेंबर दिस. देन फर्दर स्लाइड्स भी मैंने डाल रखी हैं, तो यू कैन सी दिस मेट्रो. देन अंदर कौन है? अंदर एरोबिक बैक्टीरिया. और मेट्रो में वैसे इतनी भीड़ होती है तो ये एरोबिक बैक्टीरिया चढ़ गए इसमें और वो सोच रहे हैं अच्छा अच्छा हमें तो सफोकेशन हो रही है. सो लाइफ इन अ मेट्रो जो फेमस मूवी थी वही उसी का मैंने एक एनालॉजी यहाँ पे यूज करी है. तो लाइफ इन अ मेट्रो इज टफ फॉर एरोब. सो रिमेंबर एरोबिक बैक्टीरिया आर रेजिस्टेंट टू मेट्रोनिडाजोल. एरोबिक बैक्टीरिया ऐसी मेट्रो में बैठना पसंद नहीं करेंगे, सो दे आर रेजिस्टेंट टू मेट्रोनिडाजोल. सो दिस इज द सेकंड स्लाइड. देन anaerobic bacteria are resistant to aminoglycosides. तो एनएरो में एन और अमाइनो में एम मैंने हाईलाइट किया. अनम नाम याद कर लो. तो ये याद, बस इसको ऐसे याद कर लो, एनारोबिक बैक्टीरिया आर रेजिस्टेंट टू अमाइनो ग्लाइकोसाइड्स. फिर वैंको क्या है? वैंको वर्ड का एक मतलब होता है क्रिश्चियन और इट्स ए क्रिश्चियन और हेब्रू नेम मीनिंग गॉड इज ग्रेशियस, तो दीज आर पॉजिटिव थिंकर्स. सो वैंकोमाइसिन इज रिजर्व्ड फॉर हुम? द ग्राम पॉजिटिव बैक्टीरिया. ठीक है, तो ग्राम नेगेटिव बैक्टीरिया आर रेजिस्टेंट टू वैंकोमाइसिन. रिमेंबर इट लाइक दिस. देन माय फेवरेट स्लाइड. So this is somebody going to club but could not pursue it ahead, क्योंकि गोइंग टू क्लब कॉजेज हाई एम्पियर शॉक. तो क्लब से क्या सोचोगे? क्लेप्शियला. हाई एम्पियर शॉक में एएमपी आ गया ना, एम्पिसिलिन. तो क्लेप्शियला इज रेजिस्टेंट टू व्हाट? एम्पिसिलिन. देन प्रोटियस, बर्कोल्डेरिया एंड सेरेशिया, दे आर रेजिस्टेंट टू व्हाट? पॉलीमिक्सिन बी एंड कोलिस्टिन. ठीक है. देन रिमेंबर ACE, A से अमाइनोग्लाइकोसाइड, C से सेफेलोस्पोरिन, C से एंट्रोकोकाई. तो एंट्रोकोकाई आर रेजिस्टेंट टू अमाइनोग्लाइकोसाइड्स एंड सेफेलोस्पोरिन. तो ये सारी स्लाइड्स किसकी हैं? इंट्रिंसिक एंटी माइक्रोबियल रेजिस्टेंस. ठीक है. ऑल राइट. Okay, guys. Next question. A patient presented with PUO and was suspected to have brucellosis. Serum sample was collected and standard agglutination test was performed. What is the reason for negative results in the dilutions mentioned below? So 1 into 20, 1 in 20, 1 in 40, 1 in 80 पे क्या आया? Negative result. Negative SAT. SAT by the way is the short form, accepted short form for standard agglutination test. फिर further dilutions पे test positive आया, which is the very frequently observed phenomenon in antibody detection test for brucellosis done by standard agglutination test. Now this is because of what? This is because of not antigen excess or improper reagent or invalid test. It is because of antibody excess. This is nothing but prozone phenomenon. ठीक है, this is nothing but prozone phenomenon. ठीक है. So the famous Marak's lattice hypothesis says what? Everybody should be busy. The antigen and antibody both should be busy so that a perfect lattice is formed. अगर कोई भी एक चीज ज्यादा होगी तो इम्यून कॉम्प्लेक्स बहुत छोटा बनेगा और वो प्रेसिपिटेट या एग्लूटिनेट नहीं कर पाएगा एंड वी विल मिस द डायग्नोसिस. सो इफ देयर आर टू मेनी एंटीबॉडीज देन द एक्सेस एंटीबॉडीज विल नॉट फाइंड देयर पार्टनर इन क्राइम, दैट इज द एंटीजन. सो वेरी वेरी टाइनी कॉम्प्लेक्सेस आर गोइंग टू बी फॉर्म्ड व्हिच विल नॉट बी विजिबल टू अस. ठीक है. सो वी विल मिस द डायग्नोसिस. वी विल मिस एनी विजिबल, वी विल देयर वोंट बी एनी विजिबल एग्लूटिनेशन. और अगर एंटीजन अधिक हो गया, वो एक्स्ट्रा हो गया, तो फिर उसको भी अपनी एंटीबॉडी नहीं मिल रही है. लेकिन अगर इक्विवेलेंस जोन है, मतलब एंटीबॉडी एंटीजन आर इन इक्विवेलेंट प्रोपोर्शंस, सो दैट अ परफेक्ट लैटिस इज फॉर्म्ड, देन इट विल प्रेसिपिटेट आउट और it will cause agglutination. So antibody excess is the answer over here. Next question. Which organism is transmitted by the vector shown in the image? So what is this vector? This is nothing but the trombiculate mite के larval stages. Now, so this organism, the trombiculate mite, transmits which disease? Not anaplasma or KFD or Chandipura virus. It is the causative agent of scrub typhus, which is caused by Orientia susu gamushi. ठीक है. So remember, scrub typhus जो है, it is caused by Orientia सुसु गमूशी. अब इसका यूनिक फीचर क्या है? प्रीवियसली इट वाज क्लब्ड विद रिकेट्सिए, बट नाउ इट हैज बीन सेपरेटेड फ्रॉम रिकेट्सिए बिकॉज रिमेंबर इट बिकॉज ऑफ द जेनेटिक एनालिसिस एंड ऑल्सो बिकॉज ऑफ द फैक्ट दैट इट लैक्स द लाइपोपॉलीसैकेराइड लेयर. ठीक है. एंड द वेक्टर इज, एज आई सेड, trombiculid mite, not the adults but its larvae, and the larvae are known as chiggers, and that is why scrub typhus bimari or disease is also known as chiggerosis. It is known as chiggerosis. And this is a picture of bushy vegetation which is one of the components of the zoonotic tetrad associated with scrub typhus. So bushy vegetation क्या है ये? This has got these low lying trees and bushes जहाँ पे the reservoir of this organism Orientia susugamushi they can hide. Now what are the reservoir? They are small mammals like field mice, rats वगैरह वगैरह. Now there is something called as the classic presentation or the classic triad of scrub typhus. Remember, one component is S char, ठीक है, at the site of bite by the chigger or the larva. The other is ellenpathy, lymphadenopathy, and the third component of the triad is a maculopapular rash. But this triad is seen in only 40 to 50% of the cases, and this is how the scrub typhus S char looks like. ठीक है. Then there is something called as the Susu-Gamushi triangle. Look at this. So these are the areas where scrub typhus is prevalent, and as far as India is concerned, remember, do this topic very well. इसका भी मैंने पहले एक डिटेल वीडियो करा था. I'll post the link of the same in description. This is, by the way, the most common rickettsial disease in India, and also in India it's a re-emerging disease. Please remember this. Now, what about zoonotic tetrad? Now, the first component are the trombiculate mites, that is the vector. ठीक है. The second component are what? The second component जिसे मैंने फोटो दिखाई थी आपको, bushy, the scrubby और bushy vegetation, bushy और scrubby vegetation, जिसमें कौन रहेगा? The reservoirs, that is the field mice वगैरह वगैरह, which are going to serve as the reservoir of this organism. And the fourth component is rainy or the wet season, because in the rains the chiggers are laid, they are produced. Rainy or wet season. Now इससे भी question आ रखा है, what is the drug of choice for a drug for scrub typhus? ये NEET PG का question है. Answer is doxycycline. Any alternatives? Yes, azithromycin and chloramphenicol. Chloramphenicol. Remember this. Then next question, guys. Match the following incubation period. तो कुछ भी चीज आपको इग्नोर नहीं करनी है. Incubation period पे match the following INICET में आ गया है. And recently मैंने अपने Instagram handle पर भी कुछ important diseases के incubation periods की post डाली है with some memorizing tips and tricks. Please have a look. उनके भी links मैं post कर दूंगी description में. So match the following में hepatitis A, varicella, syphilis, SARS. Hepatitis A 15 to 45 days, remember. Syphilis 9 to 19 days. Varicella का वैसे होता है 7 to 21 days, 14 to 21 days. ठीक है. SARS का होता है एवरेज 5 to 6 days, लेकिन वेरिएबल है, अप टू 14 days भी बोला गया है. ठीक है. तो वैरिसेला का हम लेके चलेंगे 10 to 20 days, एंड SARS का ले लेते हैं 1 to 10. ठीक है. सो व्हाट इज द आंसर? A3, B4, C1 and D2. This is the answer. So remember your incubation periods also pretty well. Then next question. A pregnant lady visits the antenatal clinic for her checkup. Her history is significant for having a stillbirth at 28 weeks of gestation a year back. She has had multiple sexual partners and also had a painless genital ulcer sometime before her previous pregnancy. What should be the first investigation to be done in this case? Now, so she had multiple sexual partners, also had painless genital ulcer sometime before her previous pregnancy, and she also has significant history for stillbirth. So what should we rule out in this case? All the antenatal ladies, they have to be tested for syphilis. ठीक है. So culture is not an option. Remember, Treponema pallidum, जो pathogen, it's the pathogenic strains of Treponema pallidum, they cannot be grown on artificial media. ठीक है. So HIV ELISA also not to be done here. यहाँ पे क्या करेंगे? VDRL, microscopy of sample from the ulcer also not to be done. She had a painless genital ulcer sometime before her previous pregnancy. ठीक है. तो ideally यहाँ पे हम क्या करेंगे? VDRL, because congenital syphilis also is a possibility which has to be diagnosed on time, and the treatment also has to be commenced. Next question. Protozoans seen inside the Kupffer cells of the liver: Plasmodium, Leishmania, Babesia, Toxoplasma gondii. Straightforward answer over here is Leishmania. ठीक है. Kupffer cells are nothing but the macrophages in the liver, and Leishmania is an example of an intracellular organism. Look at this. इस पे भी काफी frequently asked. This is a very frequently asked image. So this is a macrophage with numerous intracellular amastigotes measuring 2 to 4 microns in size in a GMSA stained splenic smear from a patient of visceral leishmaniasis, that is Kala Azar. Kala Azar caused by Leishmania donovani. रिमेंबर, इसके दो मॉर्फोलॉजिकल फॉर्म्स हैं. ए मैस्टिगोट जो ह्यूमंस में होता है, कहाँ पे पाया जाएगा? इन द रेटिकुलो एंडोथेलियल सिस्टम, दैट इज लिवर, स्प्लीन, बोन मैरो. एंड द प्रो मैस्टिगोट फॉर्म जो होता है वो वेक्टर में होता है, किसमें? सैंडफ्लाई में. तो एवरी दिस इज the A mastigote, this is a separate A mastigote, और जितनी बड़े आप देख रहे हो, this is nothing but the this is nothing but the macrophage filled with so many A mastigotes. So This is a single A mastigote. तो यहाँ पे आप देख रहे हो kinetoplast which is present perpendicularly to the nucleus. Kinetoplast represents the mitochondria of this organism. ठीक है? So every A mastigote has a nucleus and characteristic kinetoplast consisting of multiple copies of the mitochondrial DNA. So and this entire thing, a macrophage filled with so many A mastigotes, is known as what? It is known as LD body. LD body, not to be confused with Donovan bodies, which are seen where Donovan bodies are seen in donovanosis, which is caused by Klebsiella granulomatis, जिसका पुराना नाम क्या था? Calimatobacterium. काली माता से याद कर लो, Calimatobacterium granulomatis. Next question. The feces of cats are generally very carefully disposed of to prevent the transmission of toxoplasmosis. In which form is toxoplasmosis transmitted via cat feces? Now, cat जो है that is the definitive host and the intermediate host कौन होता है? Intermediate host is the mouse. Human beings are accidental intermediate host. Definitive host में होती है sexual cycle and oocyst is a product of sexual cycle. So the cat feces are going to have what? The oocyst. ठीक है? In the so the answer over here is oocyst. In the intermediate host, in the tissues, what is present in the tissues? The tissue cyst is present, which has got what? Which has got the slowly multiplying forms of the organisms known as bradyzoids, right? And in the intermediate host, in the blood, the rapidly multiplying forms called as tachyzoids are present. And the peculiar feature, remember about Toxoplasma gondii, is that all of these morphological forms I have listed, whether it is oocyst, tachyzoids, bradyzoids, tissue cysts, सबके सब infection cause कर सकते हैं. ये भी अपने आप में एक question आया हुआ है NEET PG में भी, INICET में भी. So infection can occur by intake or ingestion of the oocyst present in the food, by blood transfusion or transplacental transmission, in which case the infective form क्या हुआ? Tachyzoids. By intake of meat, the contaminated meat of some animal which behaved like the intermediate host, तो मीट में क्या प्रेजेंट होगा? टिश्यू सिस्ट which in turn has got bradyzoids. So ये सारे के सारे morphological forms which I've highlighted in yellow can initiate infection in humans, which are who are the accidental intermediate host. Now look at these two pictures. So these are the Toxoplasma gondii tachyzoids stained with GMSA from a smear of peritoneal fluid obtained from a lab inoculated mouse, and here we see Toxoplasma gondii cyst in the brain tissue stained with HNE. Now, next question. CDC has categorized bioterrorism agents into various categories. Which of the following statement is not true? मतलब कि वो false statement पूछ रहे हैं regarding bioterrorism agents. Category A agents spread easily associated with high morbidity and mortality. सही लग रहा है, ठीक है ये. Category B agents spread slowly associated with low mortality. ये भी ठीक लग रहा है, ठीक है. Category C agents spread easily associated with high mortality are emerging diseases. ये भी सी ठीक है. कैटेगरी डी तो है ही नहीं. री इमर्जिंग डिजीजेस हाई प्रायोरिटी, ये गलत है. गलत ऑप्शन फिर डी है. दैट इज वाई दैट इज द आंसर. दे आस्क्ड अबाउट द फॉल्स ऑप्शन. आगे देखो. दिस इज माय ओल्ड इंस्टाग्राम पोस्ट. इसका भी लिंक मैं नीचे दे दूंगी, तो यू जस्ट click on the link and save the post. So category A agents जो है ऐसे याद कर लो. ABP न्यूज सुनो ऑन टीवी. एंथ्रैक्स बॉटलिज्म प्लेज न्यूज से कुछ नहीं है, सुनो. स्मॉल पॉक्स ऑन टीवी टेलीविजन, तो टुलेरीनिया एंड वायरल हेमरेजिक फीवर्स में ये सारे आते हैं. फाइलोविरिडे, एरिनाविरिडे, बुनियाविरिडे. कैटेगरी बी एजेंट्स का नाम ही बी से शुरू होता है, तो रिमेंबर बी से ब्रूसेलोसिस. सी और डी को भूल जाओ. आजकल कॉम्पैक्ट डिस्क को यूज नहीं कर रहा है ना, आज तो स्पॉटिफाई का जमाना है, तो सी और डी कॉम्पैक्ट डिस्क को भूल जाओ. फिर डी के बाद क्या आता है? EFG. ई से एप्सिलॉन टॉक्सिन, एफ से फूड सेफ्टी थ्रेड्स, जी से ग्लैंडर्स. देन MPQRST, मेलियोडोसिस कॉज्ड बाय बर्कुलेरिया सूडोबेलाई. सेटेकोसिस, क्यू से क्यू फीवर, राइसिन टॉक्सिन, स्टेफालोकोकल एंट्रोटॉक्सिन. बी देखो यहाँ पे भी बी आ रहा है. टाइफस फीवर caused by rickettsia prowazekii. Then two more agents in category B. तो सोचो कैटेगरी बी में सबसे ज्यादा एजेंट्स हैं, बहुत ब्रॉड लिस्ट है इसकी. तो V और W याद करो. वायरल एनकेफेलाइटिस के एजेंट्स एंड वाटर सेफ्टी थ्रेट्स लाइक विब्रियो, कॉलेरे एंड क्रिप्टोस्पोरिडियम पार्वम. सो व्हाट ऑल लेटर्स आर टू बी मिस्ड इन category B? You should have clonji with double chai. This is a picture of clonji. This is a little memorizing tip I mentioned over here, ताकि आप लोगों को याद रहे. कैटेगरी सी में सी, शुक्र है इसमें सबसे कम नाम है. तो क्या देखना? नेशनल हेल्थ सर्वे पॉलिसी को देखो. तो एन से नीपा वायरस, एच से हंटा वायरस, एस से सार्स कोरोना वायरस एंड पी से पैंडेमिक इन्फ्लुएंजा. रिमेंबर दिस. Next question. A patient presented with a lesion on the left buttock. He underwent heart transplant 15 years back. Gram stain from the lesion revealed gram positive filamentous branching bacteria. What is the likely organism? gram positive, filamentous, branching bacteria. Straight away answer is Nocardia, not Mycobacterium tuberculosis or Bacillus anthracis. I always say कि clinical vignettes से बिल्कुल नहीं घबराना चाहिए. The lengthier the question, the more the hints given to you, and usually the answer is hidden in the penultimate, that is the second line, second last line of the question. Like यहाँ पे ग्रामस्टेन बता रखा है, ठीक है? So Nocardia it is. Now look at this. Nocardia on gram stain appear like these, beautifully gram positive, long filaments branching. So appearances, remember, can be deceptive. Nocardia is not what it looks like. ठीक है, कहने का मतलब क्या है? नोकार्डिया माइक्रोस्कोप में देखोगे तो लगेगा कि ये तो फंगस है, बिकॉज लॉन्ग ग्राम पॉजिटिव है, लॉन्ग फिलामेंटस है, बट एक्चुअली में ये फंगस नहीं है, ये बैक्टीरियम है. ठीक है, सो इन फैक्ट इनिशियली इट वाज कंफ्यूज्ड टू बी अ फंगस, बट later on it was found to have prokaryotic ribosomes. That is why it was labeled as a bacterium, and it responds to antibacterial agents. So these are gram positive filamentous bacteria, which are not very broad. They are very narrow, 0.5 to 1 microns wide. Remember this. ठीक है, तो नो कार्डिया, हाउ डज इट्स प्ले इट्स कार्ड्स, मतलब कि इन्फेक्शन कैसे होता है, वाया द नोज एंड द स्किन. तो दिस बैक्टीरियम कैन बी इनहेल्ड, तो वाया नोज से इन्फेक्शन हो सकता है, और बिकॉज इट्स अ सैप्रोफाइट इन द सॉइल एंड वेजिटेशन. अब ये ऐसे याद करो कि ओपनली ऐसे प्रेजेंट है, तो ये इसको तो ऑक्सीजन के प्रति एक्सपोज है, तो ये ऑब्लिगेट एरोब्स हैं. इसका जो दूसरा भाई है एक्टिनोमाइसिस, विच इज ऑल्सो ग्राम पॉजिटिव ब्रांचिंग फिलामेंटस बैक्टीरियम, वो एनारोबिक होता है. तो एक्टिनो इज एनएरो, ऐसे इट राइम्स ऑल्सो, एक्टिनो इज एनेरो. तो उसका उल्टा जो नोकार्डिया है वो ऑब्लिगेट एरोब है. ऑल्सो रिमेंबर, नोकार्डिया पार्शली एसिड फास्ट है, एक्टिनोमाइसिस एक्टिनोमाइसिस एसिड फास्ट नहीं है. ठीक है, सो वाया क्यूटिस और स्किन अगर ट्रोमेटिक इनोकुलेशन हो सकता है, दैट इज अनदर वे ऑफ इन्फेक्शन. सो दिस इज हाउ नोकार्डिया इज गोइंग टू प्ले इट्स कार्ड्स. ठीक है, नाउ लुक एट दिस गाइस. सो ग्राम स्टेन पे कैसे देखेगा नोकार्डिया? लुक एट दिस, कितने पतले पतले फिलामेंट्स हैं, ग्राम पॉजिटिव फिलामेंटस ब्रांचिंग बैक्टीरियम. ठीक है, एंड मैंने बोला कि ये पार्शियली एसिड फास्ट है. एसिड फास्ट पे ऑर्गेनिजम्स, एसिड फास्ट स्ट्रक्चर्स एंड ऑर्गेनिजम्स अपीयर पिंक, तो दैट इज व्हाई आई हैव यूज्ड पिंक कलर. यहाँ पे देखो, एंड उतने ज्यादा अच्छे से ये पिंक दिख भी नहीं रहा है, पर दिख तो रहा है. इट इज पार्शियली एसिड फास्ट. अब द डीकलराइजर विच इज यूज्ड इफ वी आर सस्पेक्टिंग नो कार्डिया when we are doing gram staining, what percentage we should use if we are suspecting nocardia? 1% H2SO4. ठीक है, so remember this, ये भी अपने आप में MCQ है. Now next question. A 25-year-old male presents with fever and severe pain in the right upper quadrant of abdomen. He had bloody diarrhea 3 weeks back. Abdominal CT scan revealed a lesion in the right lobe of the liver with ragged edges, heterogeneous densities, and multiple septations. What is the most likely diagnosis? Pyogenic liver abscess, amoebic liver abscess, hydatid cyst. The last option was not recalled. Answer over here is amoebic liver abscess. The patient has had an episode of bloody diarrhea 3 weeks back, and presentation now with fever and severe right pain in the right upper quadrant of the abdomen goes in favor of amoebic liver abscess. ठीक है, अब देखो, अमीबिक लिवर एब्सेस इज कॉज्ड बाय एंटमीबा हिस्टोलाइटिका, जिसके मॉर्फोलॉजिकल फॉर्म्स ये हैं. दिस इज द ट्रोफोजॉइड. So the pseudopodia is extension of the cell membrane which is helpful in motility. So this is the ectoplasm, and then we have got the endoplasm. Endoplasm is granular. ठीक है, RBC मैंने यहाँ पे ड्रॉ किया है. रिमेंबर, this trophozoid is said to be hematophagous. हेमाटोफेगस ट्रोफोसाइट बिकॉज इट इंजेस्ट आरबीसीज. कुछ कमेंसल स्पीशीज ऑफ एंटोमीबा भी गट में होते हैं, लेकिन वो हमारा खून नहीं चूसते, मतलब दे विल नॉट हैव इंजेस्टेड आरबीसीज, एंड दैट इज एन इंपॉर्टेंट पॉइंट ऑफ डिस्टिंक्शन बिटवीन द पैथोजेनिक एंड नॉन पैथोजेनिक स्पीशीज. प्लीज रिमेंबर दिस. तो द न्यूक्लियस हैज गॉट अ सेंट्रल कैरियोसोम एंड पेरिफेरल फाइन क्रोमैटिन. कुछ फूड वैक्यूल्स भी दिख रहे हैं, कुछ डब्ल्यूबीसीज भी हैं यहाँ पर. ठीक है, नाउ द स्कीमेटिक रिप्रेजेंटेशन ऑफ द सिस्ट. The cyst, initially the immature cyst has got a single nucleus, so this is the uninucleated cyst with glycogen mass and chromatoid body. Chromatoid body is nothing but aggregation of ribosomes, and this is the न्यूक्लियस. तो ये न्यूक्लियस डिवाइड होगा, देन वी विल गेट द बाय न्यूक्लिएटेड सिस्ट. इवेंचुअली ये दोनों न्यूक्लियाई भी डिवाइड होकर 4 न्यूक्लियाई बनाएंगे, बट बस इससे ज्यादा न्यूक्लियाई का नंबर नहीं होता है. एंड इट इज दिस क्वाड्री न्यूक्लिएटेड और टेट्रा न्यूक्लिएटेड सिस्ट विच इज द infective form. This again is an MCQ. ठीक है, this again is an MCQ. Now स्कीमेटिक रिप्रेजेंटेशन तो हो गई, इसको भी देख लो. ट्रोफोजॉइड ऑफ एंटोमीबा हिस्टोलाइटिका और एंटोमीबा डिस्पार विच आर मॉर्फोलॉजिकली सिमिलर. दिस इज इन अ डायरेक्ट वेट माउंट स्टेन विद आयोडीन. ठीक है, सो दिस इज द न्यूक्लियस, the inner granular endoplasm and the outer clear ectoplasm. ठीक है, now look at this cyst of Entamoeba histolytica or Entamoeba dispar stained with trichrome stain. There are 3 nuclei visible in the focal plane with black arrows. So black arrows nuclei की तरफ point कर रहे हैं and red arrow is showing the chromatoid body. Now look at this. Trophozoites of Entamoeba histolytica with ingested RBCs stained with trichrome stain. The ingested RBCs appear as dark inclusions. So this is also a very good picture. ठीक है, so the nuclei have got the typical small centrally located karyosome. ये रहा, this is the karyosome, यहाँ पे, ठीक है, and thin uniform peripheral chromatin. So that's about this video, guys. I really hope this video helps you. Please remember all your PYQs and PYTs. सबको अच्छे से पढ़ना. And if you have let me know in the comment section how did you like this video. मैं सारे के सारे links जिनकी मैंने बात की है इस वीडियो में मैं description में डाल दूंगी. Please share this video with your friends also so that more and more students can be benefited. And if you want me to cover any other topics, do let me know in the comment section. And my social media handles के links भी मैं description में दे दूंगी. उनको भी please देखिए, ये सारी मेहनत आप लोगों के लिए है. And if you have any doubts regarding this video, please ask your doubts in the comment section. I will be very, very happy to help. Study well, everyone. God bless. Thank you.